Okay, so gene regulation, gene expression regulation can happen in bacteria cells primarily at the transcriptional level. So it means bacteria need to be able to respond to environment pretty rapidly. For example, if they find new food, they need to be able to utilize that food for energy. So they need to be able to turn on the gene, produce a certain enzyme to be able to break down the food. So they are pretty efficient when it comes to that because they would not want to waste energy producing synthesizing enzymes that are not required for wherever they are. Eukaryotic cells, these are the cells that make up a multicellular organism, and there's a high degree of cell differentiation that's happening there to make up that multicellular organism. It means the gene expression is going to be regulated at many, many levels. Transcriptional level, post-transcriptional, translational level, and even post-translational level. We're going to explore these different levels of regulation as we move on. But first, I want to mention that genes can be inducible, repressible, and constitutive. So basically, you can turn them on, you can keep them repressed, or they can be always turned on, constitutive, meaning you have housekeeping genes, and these housekeeping genes are always on synthesizing specific proteins because these proteins are important in maintaining that homeostasis of the cell. Maybe these are structural components of the cell as well. So gene expression always begins at the promoter level, and we talked about that in a previous lecture, how transcription factors recruit RNA polymerase and initiate transcription. So we have two types of regulations here. So we have a positive regulation and a negative regulation. Positive regulation means we need an activator protein, and this activator protein will bind to the specific regulatory sequence of DNA, recruit other transcription factors, recruit RNA polymerase, and transcription is on. So you have to have that activator, so it means it's a positive regulation. Negative regulation refers to the fact that you have a repressor protein that is bound to the operator site, and because the operator is downstream of the promoter, now if you had your transcription factors and your RNA polymerase right here, that would try to move this way and read the gene. It's not going to be able to do that because that repressor protein is actually in the way. So it means no transcription is going to happen. So negative regulation means we involve that repressor to turn off the gene. So the regulatory proteins have two binding domains. So if you look at this protein right here, you're going to see there is a DNA binding domain that is going to recognize a specific binding site within the DNA. And then there is an allosteric site. So an effector protein needs to be able to bind to allosteric site that causes the conformational change in a regulatory protein. And you can see how the effector here binds to the regulatory protein, and now this regulatory protein is able to recognize a specific sequence in the DNA and turn on the gene. So this is that positive regulation with additional effector that's actually affecting the regulatory protein. You can see everything is so highly orchestrated step by step. You know, it's almost like this chain reaction. So, and if you look at the repressor, now repressor is bound to the operator, so RNA polymerase would be blocked, would not be able to get through and read the gene. However, if there is an effector, it binds to the allosteric site here. That repressor undergoes conformational change, and you can see it dissociates from the operator. So now the road is nice and clear, so it means RNA polymerase can actually read the gene and produce that mRNA molecule. Bacteria genes are grouped together. So they're not like eukaryotic ones. So this is going to be unique to bacteria. And we call these groups of genes operons. So an operon is a segment in the DNA that involves or includes a promoter, an operator, and then the actual structural gene. So these are the genes that can be expressed and then later translated into the protein. So promoter is where RNA polymerase is going to bind. That's the RNA polymerase binding site. And then the operator is the region where normally you might see a repressor sitting blocking that RNA polymerase. So that is this basic structure of a gene in bacterial cells. And yes, we can have regulatory genes, and these could be expressing specific molecules to induce and turn on the gene here. Also, what's unique to bacteria is the fact that they make what's called a polycistronic mRNA. So this is where we have one transcript, and because they have multiple genes grouped in the operon, they end up producing multiple proteins from just turning on that one region in the DNA. So because they under the same regulatory switch. So as you can see, once we have the mRNA molecule, this mRNA molecule, the transcript is going to have multiple ribosomal binding sites. So you have ribosomes that can bind here, ribosomes that can bind here in this region, and we call that region Shine-Dalgarno sequence. And let's just have fun with this. What does it read? Agagu. Agagu sounds like alien language, right? Well, that's the code. Here it is, 2 more ribosomes. So they're reading the code and then they're producing, or translation is going to be happening to produce different proteins. So you can see one transcript and then we get 3 different proteins because these genes are grouped in the operon. So one classic example in biology, whenever we talk about inducing genes, turning them on, and talking about bacterial operons, this lac operon is a prime example of how we can actually turn the genes on and off. So this lac operon utilizes lactose sugar, and if you remember from biochemistry unit, lactose is a disaccharide. So you have this galactoside linkage right here. And this is your disaccharide. You have one monomer here and here's another one. So what needs to happen? We have to break this bond and break the lactose into 2 monosaccharides. We have galactose as a monosaccharide, and also glucose right here. So bacteria needs to be able to produce an enzyme to be able to break this down, because they can't use disaccharide for the energy. So in other words, they would want to produce ATP, but they cannot utilize that directly. So they have to be in the form of monosaccharides. So this lac operon is organized, as you can see, in the following way. We already discussed that. So we have a promoter region where the RNA polymerase will bind, and then we have an operator site. This is where the repressive protein is going to be sitting at, because remember, this is an inducible operon. And then we have the structural genes. So these are going to be genes that are going to be expressed, and they're going to be translated into proteins. And these proteins will be enzymes to be able to break down the lactose. So as you can see, this gene is normally turned off because we have a repressor in a way of RNA polymerase. However, if there is lactose in the vicinity of the bacteria, and bacteria usually don't like lactose because they have to do more work to be able to get the energy out. Bacteria prefer glucose because glucose is a monosaccharide. So however, if they run low on glucose and lactose is the only nutrient that's available, they have to be able to turn on the gene and utilize that food. So lactose is going to bind to the repressive protein, and you can see repressive protein undergoes conformational change. It gets dissociated from the operator, And then RNA polymerase is now free to read the structural genes, transcribe the mRNA, which is later translated into the proteins. And there you go. We have 3 different enzymes produced and now ready to break down this. Another classic example of how bacteria can regulate the gene expression is repressible operon that we call tryptophan operon. So this tryptophan operon has genes for the biosynthesis of tryptophan, and they are clustered together under the control of that single promoter. So tryptophan is actually one of the amino acids, and you need that amino acid to be able to build proteins. So that's what bacteria is trying to do. So it's going to synthesize its own tryptophan. So therefore, this operon is always on, but it can be repressed. So now if you give bacteria tryptophan, tryptophan is going to act as a co-repressor. So it binds to the protein that is repressor, but usually it's inactive. So because obviously bacteria is trying to express a gene so it can synthesize the tryptophan. So this repressor now becomes active when the tryptophan is there. So you can see it binds to it, the repressor undergoes conformational change, and then it binds to the operator region on the DNA molecule. So now RNA polymerase, you can see right here, our RNA polymerase is bound to the promoter. However, there's a roadblock. So RNA polymerase cannot read the genes. So it means can't go this way because the repressor is in the way, and therefore no RNA made and no products produced, no proteins produced as a result. So the reason bacteria do that is because they're obviously trying to conserve energy, because if it's something that's already available in the environment, they have no need to waste energy to produce it. So this is why I put this bubble here to help you remember. Why make it if you already have it? So if you have it, you're going to turn off the genes and you're going to turn off the synthesis of necessary proteins to be able to synthesize that tryptophan. So don't waste any energy. So that is the whole idea of repressible operon.